Welcome to the Accelerated Biosciences presentation. My name is Yuta Lee, founder and CEO of Accelerated Bio. Today, we're here to talk about the ultimate breakthrough in human trophoblast stem cells, or HTSCs. Before we get started, we have an announcement to make. We're at this conference to launch our HTSC-derived iPSC cells. No human owns these ethically sourced, unique human cells. They're reprogrammed to iPSC with proprietary mRNA technology. They're protected by a robust IP portfolio. They really give you all of the business advantages and reduced business risk. Research use only is available now. GMP will be available soon. So contact us. We would love to tell you more. At Accelerated Bio, we're sourcing the highest quality immune-privileged HTSCs with no socio-ethical controversy. The technology was discovered by Professor Jianan Lee, and the company was founded in California in 2013. We're primarily focused on developing and supplying well-characterized cell types from its primary human trophoblast stem cell source. We have a broad and robust patent estate of more than 38 patents in 7 patent families. A bit about myself. I have more than 11 years experience in cell therapy development and more than 18 years in medical devices. The inventor, Professor Jianan Lee, is a physician researcher and a KOL for OBGYN in Taiwan. He got his MB from Kaohsiung Medical University, MD from Tohoku University in Japan, and PhD from St. Bart's at the University of London. We're also working with Martina Mosbergen of C14 Consulting. She has more than 25 years of business development experience in marketing and entrepreneurial experience in biotherapeutics, immunotherapies, and regenerative medicine. She has closed over $2 billion in deal value. We have a very distinguished scientific advisory board. Dr. Ben Fryer of Pluristics, Dr. Siko Patma of IO Biosciences, Dr. Peter Hudson of Cartherix, Dr. Joyce Frey Vasconcells, former Deputy Director of OTAT at the FDA, and Dr. Bram Bout, previously of Crucell and 12 years CEO of Biosciences. The main challenge today is sourcing the ideal stem cell. You're always choosing trade-offs between ethics, scale, and immunogenicity. With HTSCs, there are no trade-offs. You can have it all. Here you see a timeline of fetal development and where most stem cell sources are from. HTSCs are sourced from ectopic pregnancies between 4 to 8 weeks post-fertilization. They're still in the embryonic stage, they're immortal, and pluripotent. HTSCs are a unique and ethical stem cell source. On the left is a 5-day-old blastocyst. The inner cell mass is where human embryonic stem cells are sourced. Using HESCs destroys the blastocyst and is considered unethical. The outer ring is trophectoderm that later develops into the placenta. On the right are illustrations of an ectopic pregnancy. An embryo travels through the fallopian tube and gets stuck. It will continue to grow, and if untreated, will rupture the fallopian tube and the mother may die of internal bleeding. Typically, a woman will experience sharp pain in the abdomen, thinking it to be an appendicitis. At the ER, an ultrasound scan will reveal the ectopic pregnancy, and a salpingectomy procedure is scheduled to remove the ectopic mass to save the woman. We source HTSCs from the chorionic villi, which is the pre-placental tissue at the distal end of the non-viable fetus. Typically, an embryo will implant by day 6 post-fertilization. At 4 to 8 weeks post-fertilization, when the ectopic is discovered, the fetus is already non-viable. HTSCs can provide scale and speed with no immunogenicity. This is a table to compare some of the stem cell sources. HESCs and iPSCs are immortal and immunogenic. HTSCs can get over 75 population doublings at a 22-hour doubling time with immune privilege. As you go further down the fetal development path, you tend to lose scale and speed, but gain immune privilege. HTSCs are the earliest source of embryonic stage pluripotent stem cells with the advantage of immune privilege. HTSCs are well characterized and have signatures of both HESCs and iPSCs. HTSCs are pluripotent and can be differentiated into various cell types. We've also completed an analysis of the secretome. We also completed the HTSC-derived iPSC, creating a second platform for those looking for iPSCs. We have optimized HTSCs to scale to 85 population doublings at a 22-hour doubling time using animal origin-free, serum-free media. At 100 million cells per dose, this equals to 10 to the 28th doses. In short, one HTSC line can supply enough cells for an entire product cycle. HTSCs express HLA-G, a checkpoint molecule that keeps the fetus safe from the mother's immune system. HLA-G expression stays after differentiation and maintains immune privilege. I would like to note that it only takes one protocol step and 1 day to differentiate to neuroprogenitors and pancreatic progenitors. We have demonstrated immune privilege with 5 animal studies, all with immune competent rats. In all 5 studies, HTSC-derived cells survive the rat immune system. The cells home to and regenerate the affected organ. We did not detect any teratoma or tumor formation. We saw behavioral improvements as well. We tested a karyotype to as high as 72 population doublings. These cells are genetically very stable. We sent 9 HTSC samples to IDEXX for the H-Impact-1 evaluation. Even when the mother's blood was tested positive for CMV and HBV, all the cells were 100% negative for 25 human pathogens. HTSC as a platform has an incredibly high safety profile. We completed a proof of concept for manufacturing scale-up using bioreactors and suspension for 50 million cells. We achieved high viability and high growth. There's a lot of room for further optimization. We reprogrammed HTSCs to derive iPSCs with the proprietary mRNA technology, then differentiated them to NK cells with high cytotoxicity. So if you're looking for an NK cell platform, please contact us. We have received 38 patents in 7 patent families. This provides robust protection for the HTSC platform. HTSC is an ideal platform to accelerate the growth of the gene and cell therapy industry. We would love to tell you more. Please contact Martina Mosbergen of C14 Consulting or schedule a partnering meeting with us. Thank you for your time.